Well, what's really unique about the Tetramer assay is that it allows you to directly visualize antigen-specific T cells. The Tetramer is the main unique aspect of our assay. That's coupled with an amplification step that we do. We isolate the T cells and then we grow them in an environment that enriches the sample for the T cells that we're interested in. Then we're able to specifically label just those cells and visualize them on a flow cytometer. And so with previous assays, things like CSFE or proliferation assays that have been around for decades, what you're getting is just kind of an overall bulk measurement of what's going on with the whole T cell population. But you've got millions of T cells in the body, and they see a whole diverse set of patterns. If you're lucky, maybe 1 in 200 to 300,000 of the cells in the sample will be the one that you're looking at. And that's why this assay, it does that enrichment steps. And then with a Tetramer, because you're directly staining, you can actually visualize away from background a population that's less than 1% of the total number of cells. And so you can see very, very small differences. And you can get a fairly precise count of the number of T cells that are in that blood sample, that are in that individual, that recognize your pattern of interest. But it doesn't stop there, because the Tetramers also allow you to actually not only label those T cells, but isolate them for further analysis.
And the Tetramers that you use are actually made here at BRI, is that correct?
Yes, we're one of a very small number of labs in the country that have successfully made Class II Tetramers. Our intent is to distribute these reagents at cost to any laboratories that are interested in importing this technology.
What would you say are the most difficult or challenging aspects of this assay when it comes to replicating it in other labs?
Well, as I think about it, there's really two. The growth of the T cells is something that really is a bit of an art.